Okay, so how do we profile the morphologies? Well, when you do this many times, you of course see a vast number of cell types. And here every cell type is colored a different color. And there's many different morphologies, different ways that the dendrites are branching, different ways the axons are branching. And so you need a lot of examples of each of these cells. And here is a sort of catalog or a sort of periodic table of the main morphological cell types that there are in the neocortex. These are interneurons, they're the inhibitory interneurons. Interneurons, and these are excitatory, typically pyramidal cells, but there are also other types of cells that are not strictly pyramidal cells, but they're excitatory cells. You can see they have many different branching features. They have names, Martinotti cells, bitufted cells, double bouquet cell, bipolar cell, and so on. And these names actually come from about 100 years of anatomical study, starting with Ramón y Cajal, a very famous Spanish anatomist, who actually drew lots of cells under a normal microscope. He would actually look under the microscope and then he would just draw them. Today we can trace them in 3D, but he named many of these cells, and many of the famous anatomists that followed named many of these cells. Once you have the morphology, you can analyze this morphology, and we've released an open source tool called NeuroM, which you can actually just run and analyze these, and you can get lots of parameters about the cell, how big the cell body is, how big the arbor is, how frequently they branch, what the angle of branching is, and so on. And then these features you can use either to try to classify cells, to see which class a cell fits into, but you can also use them in other ways to build new cells. I'll show you that in a moment. So the important thing is that here is that the classification is based on local morphology. So it's just on the way that the cell looks. This, of course, is not everything about a cell. Cells may project to further places, but it's typically the excitatory cells. They project to different brain regions. They project whether it's across to the hemisphere or to deeper brain regions, or even just laterally to different parts of the neocortex. But we just look at the local morphology, and the name is based on the local morphology. Now, one of the caveats in getting morphologies in slices, brain slices, and you saw the slice before, is that you actually shave off in the slice, because this is the slice, that's the thickness of the slice. And so you shave off a part of the neuron, the branches of a neuron. So after you've done that, you have to kind of repair the missing parts. And there's software called NeuroRepair, which identifies where these cut points are, and then it grows these extended parts to be consistent with the statistics of the other side. And this is how we repair the morphologies. Sometimes we also use data from in vivo recordings to learn how to do this repairing process better. So in vivo validation. One needs a certain number of cells to be able to clone them and make a bigger data set. And here what you're seeing is the averages. The average is many cells overlaid on top of each other. And you see sort of an average axonal structure. This helps us obtain the statistics that you need to A, repair the cells. Also, you can now computationally clone these cells. You can make many copies of them within the statistical variations. They're not exact copies, they are statistical copies. So even if, in this case of the neocortical microcircuit, we have about 1,000 cells, but we have enough initial statistics, sparse data statistics, to actually build hundreds of thousands or millions of copies of each of these different cell types. So you can produce many, many copies. That's one approach, and a more recent approach that we're using, and we won't go into that in this MOOC, but is where we actually computationally synthesize them using the rules of how they branch. And the idea here is that you want to learn how to synthesize any cell type in the brain. And that's a long-term goal.